Usually when we talk about human anatomy, there is this expectation of, like, solid, visible architecture.
Right, like a building or a machine.
Exactly. You look at a skeleton, you see the femur, and you can point right at it and say, well, there it is. That is what holds us up.
It feels permanent. I mean, it's comforting, really, to our macroscopic brains. But when you actually zoom in past the bones, past the organs, that solid architecture kind of just melts away entirely.
Yeah, because if you step into the realm of physiology, you are looking at a landscape that is mostly just fluid, shifting pressures, and, invisible electrical storms. Everything we consider solid is really at the mercy of these microscopic gradients.
It is. And that transition from the gross anatomy lab to the microscopic fluid dynamics, well, it's usually where people get lost.
And that is exactly our mission today. We are acting as your personal medical experts, basically ripping open the textbooks, specifically the USMLE Step 1 Physiology notes from 2021 to decode this liquid machine for you.
We want to break down how these vital physiological systems actually work, translating dense medical mechanics into practical working knowledge.
Okay, let's unpack this. Because before we can even talk about the major organs, we have to talk about the water. I mean, about 60% of your total body mass is literally just water. It's the foundation of everything.
But that water isn't just sloshing around randomly either. The body meticulously divides it into compartments using these highly selective membranes.
Right, like dividing a ship into watertight rooms.
Exactly. The cell membrane, for instance, acts as a relative barrier keeping sodium out, while the capillary membrane acts as a barrier keeping larger things like plasma proteins trapped inside the blood vessels.
The movement of fluid between these spaces is entirely governed by osmosis. My understanding of osmosis is that water, it always wants to move from where it's highly concentrated to where it's less concentrated. Or, more simply, water chases solutes.
It does. It always moves toward the higher concentration of particles. That solute-chasing behavior is why we measure osmolarity in milliosmoles per liter. It's strictly about the actual number of particles in the solution.
No, it doesn't matter what the particle is.
Not for osmolarity, no. If you trap a high number of particles on one side of a membrane, the water molecules will literally pull themselves across that barrier to dilute the crowded side. It is just a relentless physical force.
Which brings us to the specific ions dissolved in that water. Because inside our cells, we have a very high concentration of potassium, along with these large, negatively charged proteins that are physically trapped inside.
Yeah, the proteins are too big to leave.
Right. But outside the cell, in the extracellular fluid, there is a very high concentration of sodium chloride and calcium.
And because of that specific unequal distribution, we get what is called the membrane potential, or EM.
EM.
Yeah. In excitable tissues, at rest, there is a clear separation of charge across that membrane. The inside is negative relative to the outside, sitting roughly at negative 70 millivolts.
Okay, so are cells essentially tiny, perfectly balanced batteries filled with seawater just waiting to discharge? Because we have this electrical gradient, like charges repel, opposites attract, and we have a chemical concentration gradient pushing these ions around.
Well, what's fascinating here is I'm actually going to push back on the phrase perfectly balanced.
Oh, really?
Yeah, it's actually the exact opposite of balance. It is deliberately, violently unbalanced.
Wow. Violently.
Truly. The cell expends massive amounts of energy using the sodium-potassium pump to force sodium out and pull potassium in, specifically to create this precarious state. So instead of a battery, think of it like a massive dam holding back a highly pressurized reservoir.
Okay, I like that analogy better. So the sodium is sitting outside, positively charged, just staring at the negative sodium-depleted interior of the cell, basically desperate to flood in.
Exactly. And that negative 70 millivolts represents the potential to do work. But to do work, you need flow.
Right, you need to open the dam.
Yes, and that's where the concept of conductance comes in, which is represented in physiology by the letter G. Conductance is all about the open or closed states of ion channels in the membrane. They are the floodgates of your dam.
So when a channel opens, the membrane's permeability to that specific ion increases.
You open them, and the ions violently rush in based on the net driving force.
And the net driving force is just the mathematical difference between the membrane potential and the ion's equilibrium potential, right?
Exactly. It essentially quantifies how far that ion is from being perfectly balanced, which dictates the force pushing it across the membrane. That sudden rush of ions, that discharge of the cellular dam, is the action potential. It's the electrical spark that tells a muscle to move.
Wait, so we have this microscopic spark, but an electrical spark doesn't lift a box? How do we get from a rush of invisible sodium ions to actual physical force?
Well, to understand how that spark generates tension, you have to look at the mechanical loads placed on a muscle, specifically preload and afterload.
Preload and afterload. Okay, let me see if I can visualize this. Preload is the load on a muscle when it's in a relaxed state, right? Like before it actually contracts.
Yes, exactly.
So when you apply preload, you stretch the muscle and you stretch its microscopic functional units, the sarcomeres. And because muscle is elastic tissue, it naturally resists being stretched. This generates what the book calls passive tension. The greater the preload, the greater the stretch, and the greater the passive tension.
Spot on. Then you have afterload. Afterload is the actual physical force the muscle must work against when it attempts to contract. If you're trying to pick up a 10-kilogram weight off the floor, that 10 kilograms is your afterload.
So preload is like pulling back a rubber band, creating that natural snapback tension, and afterload is the literal physical rock you've placed in the band trying to launch it.
That holds up perfectly. When the action potential fires, the muscle generates active tension.
Active tension.
Right. On a microscopic level, this is myosin protein heads grabbing onto actin filaments and physically pulling them, which is the cross-bridge cycle. Your total tension is just the sum of that active tension from the pulling plus the passive tension from the stretched preload.
But whether that rock actually flies depends on how much force those proteins can generate against the weight. The source text makes a big distinction here between isotonic and isometric contractions.
It's a crucial distinction.
Yeah, so if the muscle generates more than 10 kilograms of force, it overcomes the afterload. The muscle shortens, the weight moves. That is an isotonic contraction.
But if the muscle generates less than 10 kilograms of force, it cannot overcome the afterload. The muscle builds up tension, but it cannot shorten.
So the weight doesn't budge.
Right. That is an isometric contraction.
So if I'm holding a heavy box and my arms are trembling, but the box isn't going anywhere, that's isometric.
It is. But if we connect this to the bigger picture, the fascinating part isn't just the lack of movement. It's that on a microscopic level, those cross bridges are still furiously cycling. They are grabbing, pulling, burning massive amounts of cellular energy ATP. But the external load is too heavy for the muscle fibers to physically shorten.
Wow, so you are exhausting the muscle without moving an inch.
Exactly, which raises a huge logistical problem for the body. If these muscles are furiously burning ATP to generate all this tension, they need a constant, massive delivery of oxygen and nutrients. A single cell can rely on simple diffusion, but a massive bicep.
That requires a dedicated bulk flow delivery network operating under its own constant pressure.
Enter the cardiovascular system, the ultimate pump. It is essentially a continuous whole-body cycle of preload and afterload moving fluid through a closed loop.
And consists of two pumps, right? The right and left ventricles and two circuits. The pulmonary circuit sends blood to the lungs, and the systemic circuit sends it to the rest of the body.
Yes. And because these two circuits are connected in series, meaning one feeds directly into the other, the flow must be perfectly equal. The cardiac output of the right side has to match the cardiac output of the left side, down to the milliliter over time.
Because otherwise blood would instantly pool and back up in the lungs or the systemic veins.
Exactly. So the blood leaving that left heart enters the systemic circuit. It goes into the aorta, then branches extensively down into the arteries, the arterioles, the capillaries for exchange, and then gathers back up into the venules, veins, and vena cavae to return to the heart.
The structural MVPs here seem to be the arterioles, right?
Without a doubt. The muscular arterioles are the primary site of resistance in your entire body. If we sum up all the resistance from every arteriole, we call that total peripheral resistance, or TPR.
And linking this back to our muscle mechanics, this TPR represents the afterload to the heart.
Exactly. It is the literal physical resistance the left ventricle must push against every single time it beats.
So there has to be a mathematical relationship here between how much blood the heart is pushing out and how much the arterioles are pushing back.
It's one of the most vital equations in all of physiology. I mean, arterial pressure, or MAP, equals cardiac output multiplied by total peripheral resistance. MAP equals CO times TPR.
Okay, MAP equals CO times TPR.
Right. The arterioles essentially function like faucets, and the body has two competing needs.
Yeah.
Locally, The tissues need to regulate these faucets to ensure they get enough oxygen. That's intrinsic regulation. But systemically, the whole body must maintain a high enough upstream pressure, that mean arterial pressure, so that blood can reach the top floor.
Meaning in the brain.
Right. That's extrinsic regulation.
Wait, here's where it gets really interesting, and I have to push back a bit. If every organ needs blood, and they all have the ability to organically open their own local vessels, wouldn't a sudden full-body demand just crash the entire system?
How do you mean?
Well, if I go for a dead sprint, my leg muscles demand oxygen, my heart demands oxygen, my lungs are working. If they all twist their local arterial faucets wide open simultaneously, the pressure in the pipes should drop to a trickle. How does the brain not get starved of blood every time we exercise?
It's a terrifying scenario, honestly, and it's exactly why extrinsic regulation overrides local demands. To prevent a total pressure collapse, the body relies on the sympathetic nervous system, the SNS.
The fight or flight system.
Yes, but even when you are completely at rest, the SNS is the dominant regulator of vascular tone. It constantly drips norepinephrine onto alpha-1 receptors on the smooth muscle of those arterioles.
So it's always squeezing them a little bit.
Exactly. This causes constant baseline vasoconstriction. It intentionally keeps the faucets partially closed to maintain that essential MAP.
And the body is constantly monitoring this upstream pressure to make sure it doesn't dip. We have these cardiopulmonary and echinoreceptors, baroreceptors, embedded directly in the walls of the heart, the great veins, and the pulmonary artery.
They act as volume sensors.
Right. If fluid volume drops, they immediately fire off a reflex to the medulla and the brainstem via the vagus nerve.
And that reflex instantly ramps up sympathetic activity, squeezing the vessels tighter. It decreases parasympathetic activity to let the heart rate spike, and it stops the inhibition of antidiuretic hormone, telling the kidneys to hold on to water. It is a desperate negative feedback loop to save your blood pressure.
This isn't just theory either. The source notes that conditions like sepsis, anaphylaxis, or neurogenic shock involve uncontrolled massive vasodilation.
Right. The local faucets are thrown wide open against the body's will.
And mean arterial pressure plummets.
Which is why in emergency pharmacology, if a patient is in distributive shock, we bypass the failing nervous system entirely. We administer synthetic drugs that mimic norepinephrine. We give massive doses of alpha-1 agonists to force those smooth muscles to constrict and artificially restore the pressure head before the brain loses perfusion.
Okay, so the heart and the nervous system are violently protecting this high-pressure systemic system. But where is all this pressurized fluid actually going? It's going to the organs of exchange. Let's look at the lungs first.
Yeah, the whole point of pumping blood through the pulmonary circuit is gas exchange. We measure the oxygen content in that blood in volumes percent. Typically, healthy systemic arterial blood holds about 20 volumes percent, meaning 20 milliliters of pure oxygen gas dissolved in every 100 milliliters of blood.
But when you look at the exchange process itself, it's not a perfect transfer. The partial pressure of oxygen in the alveoli, the tiny air sacs in the lungs, is roughly 100 millimeters of mercury. But the oxygen pressure in the systemic arterial blood leaving the lungs is slightly lower, sitting around 95.
And this creates what's called the A gradient, the alveolar to arterial gradient. In a healthy young person, that gradient is incredibly small. But clinically, it's vital to understand that this gradient naturally widens as we age.
The source actually provides a formula to estimate a normal A gradient for any patient.
Yes, you take their age, add 4, and divide by 4.
But why does that happen? I mean, why does a 70-year-old naturally have a harder time moving oxygen from their lungs into their blood than a 20-year-old?
It comes down to microscopic wear and tear. Over decades, the lungs lose some of their natural elasticity. Microscopic scarring occurs, and the delicate matching of airflow to blood flow, what we call ventilation perfusion matching, gets slightly misaligned.
So gravity affects the lower parts of the aging lung differently.
Exactly. So even if the alveoli are full of oxygen, the blood flowing past them isn't capturing it as efficiently. If a patient's gradient is significantly larger than that mathematical formula predicts, it points to a serious underlying pathology causing hypoxemia, like a pulmonary embolism or severe fibrosis.
Meanwhile, as the lungs are handling gas exchange and blowing off CO2, the kidneys are handling the long-term regulation of our fluids, acid-base balance, using the CO2 bicarbonate buffer system. But the kidneys are also filtering massive amounts of this fluid. And I want to point out a fascinating contradiction here. We just talked about the heart fighting tooth and nail to maintain high systemic blood pressure. But the source points out that the kidneys essentially install their own massive pressure regulator. They autoregulate their glomerular filtration rate, or GFR, and their renal blood flow.
They do.
Do the kidneys just ignore the cardiovascular system's pressure swings as long as MAP is between 80 and 180?
They have to. Without that autoregulatory range, the capillaries inside the glomerulus, the filtering unit of the kidney, would be destroyed. They're incredibly delicate. If they were subjected to every single spike in systemic blood pressure when you exercise or get stressed, those capillaries would burst, leading to permanent kidney damage.
Oh, wow.
So the kidney actively changes the diameter of its own entrance vessel, the afferent arteriole. If systemic pressure spikes, the afferent arteriole clamps down, shielding the glomerulus from the pressure wave. This ensures that no matter what the heart is doing, the filtration rate stays perfectly flat and stable.
That is an incredible localized defense mechanism. And we can actually measure how much water these kidneys are reabsorbing by looking at a substance called inulin.
Ah, yes, inulin.
It's a brilliant reference marker because it is freely filtered by the glomerulus, but once it's in the kidney tubules, the body doesn't reabsorb it and it doesn't secrete any extra into the tube.
Imagine a microscopic pipe where the fluid is flowing through. The cells lining the pipe are furiously sucking water back into the body to prevent dehydration, but the inulin particles are entirely trapped in the pipe. As the water volume drops, the remaining inulin particles are forced to crowd together, increasing their concentration.
So if you look at the concentration of inulin in the proximal tubule fluid compared to the blood plasma, the TFP ratio, and the source says it's 3.0, that tells you the story of the water.
Right.
Since the inulin concentration tripled, the volume of water it's dissolved in must have shrunk to 1/3 of its original size. Therefore, 2/3 of the water was reabsorbed back into the body.
Precisely. It allows us to calculate exactly what the kidney is saving versus what it is discarding. And speaking of discarding, the way fluid finally leaves the body, the micturition reflex, or urination, is a masterclass in how the autonomic and somatic nervous systems hand control back and forth.
The filling phase is entirely dominated by the sympathetic nervous system, right? The same system keeping our blood pressure up is also acting as the storage system for urine.
Yes. While the bladder is filling, sympathetic nerves release norepinephrine. This hits beta-3 receptors on the detrusor muscle of the bladder wall, commanding it to relax so the bag can expand.
And at the same time,
Simultaneously, it hits alpha-1 receptors on the internal sphincter, commanding it to tightly pinch the exit shut, keeping the fluid inside.
but the voiding phase switches over to the parasympathetic system. As the bladder stretches, it sends sensory signals to the spinal cord. When it's time to void, the parasympathetic system kicks in, releasing acetylcholine.
And this hits M3 receptors on the detrusor muscle, commanding the bag to vigorously squeeze.
Meanwhile, the external sphincter is under our voluntary somatic control via nicotinic receptors. We actively send signals from our brain to keep it contracted until we decide it is socially appropriate to relax it and allow emptying.
Exactly.
So we've seen how the nervous system acts with lightning speed to change blood pressure and how the kidneys filter that fluid. But the body also relies on a slower, highly localized chemical regulation system to manage the cellular energy dissolved in that fluid, specifically glucose. Enter the endocrine pancreas.
The endocrine functions of the pancreas are localized in tiny, highly organized clusters of cells called the islets of Langerhans. And these islets contain very specific hormone-secreting cells.
You've got your beta cells making up the vast majority, about 60 to 75% of the islet, and they make insulin. Then you have alpha cells, which make up about 20%, making glucagon.
And finally, delta cells.
Right, making up just 5%, which secrete somatostatin. But what stands out in the source notes isn't just the percentages, it's the physical architecture. The alpha cells tend to be located on the outer periphery of the islet, while the beta cells are clustered densely in the center.
And this is where form perfectly dictates function. This raises an important question about how they communicate. The blood supply to the islet doesn't just wash over it randomly. Blood flows specifically from the center of the islet outward to the periphery.
From the center out.
Yes. So the blood first washes over the beta cells in the center, picking up a massive concentration of insulin. Then, carrying that high concentration of insulin, the blood flows outward to the alpha cells on the periphery. This literal microscopic plumbing allows the insulin to directly wash over the alpha cells and inhibit the secretion of glucagon.
So what does this all mean? The architecture of the pancreas is the communication system. The literal direction of blood flow determines which hormone shuts off the other.
Exactly. If the blood flowed the other way, the entire metabolic balance of the human body would be inverted. It highlights that physiology isn't just a soup of chemicals, it's highly directional. And there's one more vital piece to this insulin story. When those beta cells synthesize insulin, they don't just manufacture the active hormone, they synthesize a large precursor molecule called preproinsulin.
Why build a massive precursor just to cut it up? The source notes it gets cleaved into proinsulin, and then before it is finally secreted, proinsulin is cleaved one last time. It physically splits into equimolar amounts of active insulin and a connecting peptide known as C-peptide.
Right, for every one molecule of insulin released into the blood, exactly one molecule of C-peptide is released.
Right.
Historically, C-peptide was viewed as biological trash, just a useless byproduct of cutting up the proinsulin molecule.
But that's changed, right?
Drastically. Clinically, because endogenous insulin and C-peptide are secreted in that exact 1-to-1 ratio, C-peptide serves as an incredibly useful long-term marker. If a patient is taking synthetic insulin injections, measuring their blood insulin doesn't tell you what their own pancreas is doing.
Oh, because you can't tell the synthetic from the natural.
Exactly. But measuring C-peptide tells you exactly how much native insulin their beta cells are still managing to produce.
And it turns out it's not just a marker. Modern research, highlighted in the text, suggests that C-peptide receptors actually exist on various cells. It is now believed to have a protective biological role, helping to prevent the renal, neural, and microvascular damage we frequently see in type 1 diabetes, where C-peptide is entirely absent because the beta cells are destroyed.
In physiology, there really are no useless byproducts. Everything eventually reveals a purpose. It's a vast interconnected web where changing a single variable, a single resistance vessel, a single ion channel, or a single cleaved peptide cascades through the entire body.
Okay, let's take a breath and recap the incredible journey we've just been on. We started at the absolute microscopic foundation, the physics of water osmosis, and the deliberately unbalanced, highly pressurized electrical dams of our cell membranes.
We scaled up to see how those voltages trigger the cross-bridge mechanics of muscle tension.
We watched the ultimate muscle, the heart, pump that fluid against the systemic resistance of our arterial faucets. We filtered that fluid through the lungs and the precise auto-regulating pressure valves of the kidneys. And finally, we explored how the literal directional plumbing of the pancreas controls our metabolic hormones.
It is an elegant, relentless machine.
Now, because our mission today is educational, we want to challenge you, the listener, with a specific review exercise to test your integration of these concepts.
Time for a little pop quiz.
Exactly. Based on everything we've covered today, if a patient experiences severe blood loss, say, from trauma, which drastically reduces the fluid returning to the heart and drops their preload, how exactly do the cardiopulmonary mechanoreceptors alter the sympathetic nervous system activity and those arterial faucets to maintain mean arterial pressure? Think through the steps, the specific receptors involved, and the math of MAP equals CO times TPR.
And while you're working through that pathway, I'll leave you with one final provocative thought to ponder something that builds on the respiratory material we discussed. We learned today that the AAA oxygen gradient naturally increases using the formula H plus 4 divided by 4.
Right.
I want you to think about what that implies for human physiology over a lifespan. Does normal aging fundamentally mean our body is slowly, inevitably losing its physical ability to extract the very gas that powers our cellular batteries?
That is a profound question to leave on. Thank you so much for joining us on this deep dive. Your dedication to mastering the complex, beautiful, and sometimes murky machinery of the human body is what makes this exploration so incredibly rewarding. Until next time, remember the solid structures might give us our shape, but it's the invisible fluid dynamics that truly keep us alive. Keep learning.